Hello, everyone. My name is Amber Royster, and I am going to discuss a research article from Nature titled The Person-to-Person Transmission Landscape of the Gut and Oral Microbiomes. This is a study focusing on the transmission of microbes represented by the rate of shared strains between individuals and how these rates correlate to the structure and composition of the human microbiome. I found this paper to be an insightful look into how our microbiome is shaped by our everyday interactions with other people. I hope you find the study as interesting as I did, and with that said, let's get started. So the bottom line up front. The intent of this paper is to analyze how members of the microbiome are acquired and transmitted, and how this affects the composition of the microbiome as a whole. The researchers wanted to better understand the modes and pathways of the microbiota transmission and what the impact of that transmission was on both an individual and a societal level. The study sought to track the microbial transmission between individuals by observing shared strains between individuals. Studying the microbiome can help us understand how microbiomes are constructed, how they adjust over time, and how they contribute both to health and to disease. We interact with microbes every day, whether it's from touching things or from being around other people. And as humans, we are social beings, and that means consistent contact with others. By interacting with other people, we are constantly introducing new bacteria into our bodies and integrating them into our microbiomes. Many studies have been conducted on the transmission of microbes, but not many have focused on human microbiota. Most of the research focus has gone to the transmission of pathogenic bacteria, and rightfully so. Commensal bacteria has been comparatively ignored. Introducing new bacteria into our microbiomes could have very significant impacts to our health, and it is vital to understand the transmission of these microbes. In this paper, the main variable measured by the researchers to determine transmission is strain sharing. Strain sharing is the transmission of genetically unique microbial strains between individuals. By detecting the same strains within two people in a given area, it would suggest that perhaps a transmission event occurred between those two individuals, or perhaps they both acquired the strain from a shared environment. Nevertheless, monitoring the shared strains in individuals can assist in understanding transmission. Strain sharing is measured by using a strain sharing rate. That is, the total number of shared strains between two individuals divided by the total number of shared species-level genome bins, which are groups of metagenome assembled genomes that represent microbial species. This fraction is then multiplied by 100 to generate a percentage. For this slide, I wanted to provide a quick overview of the two microbiomes covered in this study. The first microbiome discussed in this paper is the gut microbiome. We are all familiar with the many vital functions that the gut microbiome provides, such as digestion, immune function, as well as neurological mediation via the gut-brain axis. The microbiome is established early in life via vertical transmission at birth. In addition to other people, gut microbes can be transmitted by water, food, and the environment. The second microbiome discussed in this paper is the oral microbiome. This microbiome also has a significant impact on our health, especially our systemic health as well as our oral health. In comparison to the gut microbiome, the oral microbiome is much more communicable and therefore much more easily transmitted. Both of these microbiomes are very diverse, as I have described in this slide, and are excellent microbiomes to monitor transmission. For the study's methodology, over 9,000 samples were evaluated across 31 different datasets, 8 of which were novel to this study. The breakdown of the relationships tested in this study is represented by this transmission set graphic from the paper. Mother-offspring pairs and household inhabitants were sampled for both the gut and oral microbiomes, while the gut microbiome had additional datasets from sets of adult twins. These samples were sequenced using 3 different Illumina platforms, all of which are well suited for large-scale microbiome studies. The sequence data was then categorized into the previously mentioned species-level genome bins, or SGBs, and taxonomically assigned. From here, strain profiling was obtained and the strain sharing rates could be calculated. Here is the workflow of the experiment. The sample datasets were compared to an expanded SGB database using a species profiling computational tool, which allowed for the samples to be profiled on a species level. The samples were then profiled on a strain level analysis using a strain-specific computational tool. After strain profiling, the researchers removed putative food strains from the data. These food strains are bacteria that are commonly found in commercial fermented foods. Since these strains are commonly found in food and could be introduced into the human microbiome that way, they were removed as to not skew the person-to-person transmission rates being monitored in this study. The remaining strains were then ran through phylogenetic distance thresholds in order to determine any strain sharing event. Strain sharing was then assessed and the strain sharing rates were calculated. Now for the results and findings of this study. The graph on the slide represents the gut microbiome strain sharing rates for each relationship noted on the x-axis. Each circle represents one strain sharing rate between the individuals within the relationship. The researchers used the range of strain sharing rates within each relationship to generate a box plot, which represents the range between the lower and upper quartiles. The line in the middle represents the median. The first set of data on the graph represents strain sharing within the same individual. These individuals were tested twice 6 months apart in order to generate this data. The median strain sharing rate for between the same individual was 87%. The next set of data represents shared strains between mother and offspring, which we'll talk much more extensively about in the next slide. At a median of 34%, mothers and offspring had the highest rate of shared strains. This could partially be due to cohabitation, but a larger factor in the pairing is the vertical transmission from mother to offspring. This is the only relationship in this data able to have this transmission pathway, so it must be taken into consideration. Also, in this particular graph, offspring refers to children aged 3 years or younger. The next group measured is household members, which includes children above the age of 3. The median strain sharing here is 12%, significantly lower than the mother-offspring median rate. Next are non-cohabitating adult twins, both dizygotic and monozygotic, which at 8% had a slightly lower rate than household members. Non-cohabitating adults in the same village also had a median strain sharing rate of 8%. Non-cohabitating individuals in different villages within the same population-specific study cohort, which is represented by the within population designation, and between different population-specific study cohorts, which is represented by the interpopulation designation, both had medians of 0%. All comparisons are statistically significant with a p-value of less than 0.05. The numbers along the top of the graph represent the percentage of pairs in each relationship that had no strain sharing. So, while only 4% of the mother-offspring pairs had no shared strains, 97% of pairs in the interpopulation designation had no strain sharing. These results displayed both a social distance bias and a familial bias in transition rates. Those in more frequent and constant contact with each other have higher rates of strain sharing, as do those with close kinship, such as mothers and offspring. One of the major relationships observed in this paper was the transmission between mothers and offspring. As represented in the graph, in the first year of life, the strain sharing rate is relatively stable, with 60% in the first day of life, 50% in the first week, and 47% in the first year of life. However, we see a significant decrease in strain sharing to 27% as the offspring ages to 3 years. This downward trend continues and stabilizes as offspring age throughout childhood and adulthood. Interestingly, there is a slight uptick in strain sharing at the 85-year-old mark. Also plotted in this graph, represented by the dashed line, is the average number of species-level genome bins, or SGBs, found in the offspring. In other words, this line represents the number of unique species detected within the offspring's microbiome at each age interval. In the first year of life, the offspring has very few diverse species in its microbiome, but as the age of the offspring approaches 3 years, we can observe a significant increase in the average SGB count, and this continues throughout childhood. Notably, the average SGB decline, approaching 85 years for the offspring. So we have a negative correlation between the strain sharing rate between mother and offspring and the number of diverse species within the offspring's microbiome. As the offspring's age increases, so does its number of microbial species, while the maternal strain sharing rate decreases. This is most likely due to the offspring becoming less dependent on the mother as they age, including socializing in other environments where the mother is not present. From the graph, it's clear that early colonization of the gut microbiome is important and lasts even as the individual approaches elderly age. Maternal transmission in general sets the foundation of the offspring gut microbiome. It is also clear that the offspring horizontally acquires microbes from non-maternal sources as they age. There is one other thing I would like to mention about the data in this article. Every circle plotted on this graph is representative of a mother-offspring pairing, and each color corresponds to that pair's respective dataset. As previously mentioned in the methods section, there were 31 different datasets analyzed for this study. As you can see, different datasets were analyzed for different ages. For instance, the purple data points towards the left-hand side of the graph correspond to a dataset from Italy and are only relevant to the offspring's first day, week, and year of life. Conversely, the green data points at the end of the graph correspond to a dataset from China and are only relevant to the data for the 85-year-old offspring. While having a varied dataset with a global array of samples is valuable, and the resulting data provides insight into transmission between multiple interpersonal relationships, I thought it was worth mentioning that there are very diverse datasets used for this analysis, and that there could be both environmental and lifestyle factors that are unaccounted for. To determine the effects of vertical transmission on the gut microbiome, strain sharing rates between mothers and offspring were evaluated, focusing on the comparison between mothers with vaginally delivered offspring and mothers with cesarean delivered offspring. In this graph, we can see that the vaginally delivered pairs had a markedly higher strain sharing median rate than their cesarean counterparts from the first day to the first year of the offspring's life. The biggest gap is at the year mark, where there is a difference of about 50% between the two groups. However, at 3 years of life, we can observe a shift in the median rate, with cesarean pairings having slightly more shared strains. This shift increases further at 18 years. This decrease in strain sharing further proves that, as the offspring age, they are introduced to new microbes and lose microbial similarity with their mother. Strain sharing rates between mothers and offspring is at its highest with vaginally born offspring in the first year of life, proving that early colonization of the infant gut microbiome is largely influenced by the vertical transmission from its mother. To evaluate horizontal gut microbiome transmission, the researchers studied individuals from both rural and urban households with varying levels of industrialization. Most of the households display substantially higher strain sharing rates among cohabitating members than with non-cohabitating people of the same population. The researchers then evaluated strain sharing between different familial relationships of household members. This is represented on the graph on the left. For this graph, the offspring studied were 4 years of age and older. Maternal and paternal strain sharing rates with offspring have similar median rates and overall ranges, with mother offspring rated slightly higher, most likely due to vertical transmission. Interestingly, sibling strain sharing rates are slightly higher than both parental rates. This could be due to both siblings' microbiomes being young and therefore simple in their composition, but there also could be a genetic component constituting the difference. There's also the possibility that, as siblings in close age, they are exposed to the same non-parental microbes from other environments and have been similarly colonized. In order to measure the effect of cohabitation on strain sharing in adulthood, pairs of adult twins were studied and compared, and this is represented in the graph on the right. These twins, which included both dizygotic, which is fraternal, and monozygotic, identical, twins, were currently non-cohabitating at the time of the study, but had lived together in the past. The rates of strain sharing between twin pairs decreased significantly the longer the twins had lived apart. This also correlates to the previous point made about the increasing microbial diversity as time moves on and as the individual ages. Both non-cohabitating twins had developed separate gut microbiome compositions. It was noted that the monozygotic twins had, on average, higher strain sharing rates compared to their dizygotic twins, suggesting a genetic component to gut microbiome transmission. Now we turn our attention to the oral microbiome. Although the oral microbiome is more easily accessible and therefore more transmittable than the gut microbes, person-to-person transmission of the oral microbiome is still largely a mystery. On the left graph, we have another box plot detailing the strain sharing rates of oral microbes. Here we can see that all three cohabitating relationships, so the mother to offspring relationship, the father to offspring relationship, and the partner relationship, all have significant rates of shared strains. In fact, the median strain sharing rate between all three cohabitation relationships is 32%, while the within population and interpopulation relationships have 3% and 0% of shared strain rates, respectively. This means that cohabitating relationships display 10 times higher oral strain sharing rates than non-cohabitating relationships. Again, looking at the numbers along the top of the graph, we see that all cohabitating relationships have less than 1% of pairs with no strain sharing event. And with partners, we see that there were no pairs that did not have any shared strain. On the contrary, we have the interpopulation designation, which has the majority of their pairs with no shared strains. Perhaps the most interesting data with the oral microbiome is the strain sharing rates between mother and the offspring and father and offspring, plotted against the age of the offspring, represented in the graph on the right. If you recall, the gut microbiome graph detailing the mother offspring rate over the offspring's life, the strain sharing rate decreased as the offspring's age increased. Here with the oral microbiome, we see the opposite trend. As the offspring gets older, the strain sharing rate increases with both the mother and the father. Also, if you look on the right side of the graph, we can see the number of SBGs in the offspring's oral microbiome drastically increases as well, as that dashed line there. So the number of SGBs in the offspring's oral microbiome increases as well, which is in line with what the gut microbiome did. So, what do the results mean? For starters, cohabitation is the biggest factor in the personal genetic makeup of the human microbiome. Long-lasting close contact is proven to be the most substantial parameter in microbiome transmission and colonization and provides the highest metabolic potential. Both the gut and the oral microbiomes showed significant similarity between cohabitating individuals. That said, The effects of cohabitation can be partially reversed with time, distance, and age, as individuals are introduced to new environments and new people. Social interactions are highly influential to our microbiome and could possibly play a role in diseases associated with their microbiome. As predicted, strain sharing between mother and infant had the greatest rates, especially in the first year of life. This drives home that vertical transmission is fundamentally important to the foundation of the human microbiome. Lifestyle had very little effect on microbial transmission. This study compiled data from multiple different countries, both westernized and non-westernized, and both the vertical and horizontal rates were similar in each group. Person-to-person transmission impacts us at our very start of life and continues to impact us throughout the entirety of our life. That said, our interactions with other people during our lives greatly changes our microbiome, introducing new species and increasing our microbial diversity, which is necessary for a fully functional microbiome. There is one limitation that I found in this paper. This study only focuses on the gut and oral microbiomes and excludes other human-associated microbiomes that are susceptible to colonization through transmission. For instance, the organ that comes into the most contact with the environment is the skin. So the skin microbiome could have been insightful in the study. The nasal and lung microbiomes are also heavily associated with the outer environment and could shed light on airborne transmission. Future transmission studies should incorporate these microbiomes. Also, with so much focus on familial relationships in this study, genetics seems to have strong influence in transmission and colonization of the human microbiome. The connection between microbe transmission and individual genetics should be further studied. The shared strains between family members is influenced by cohabitation, but these family members could also have a genetic predisposition to these microbes. Further studies should be conducted to outline genetics' role in microbial transmission. Okay, so some questions to consider. First question, why do you think this study focused on just the gut and oral microbiomes? And should similar studies be conducted on other human-associated microbiomes? I mentioned in my conclusion section that the skin, nasal, and lung microbiomes are all susceptible to colonization from transmission. Do you believe it would have been beneficial to analyze these microbiomes or any other human microbiome? Or do you think the gut and oral microbiomes were chosen for a specific reason? Question 2. How large a part do genetics have in microbial transmission and colonization, and do you believe more research should be done to explore this connection? So we've seen in this study that familial relationships have the highest rates of strain sharing. Is that solely because of cohabitation and early colonization, or is it possible that the similar genetics between family members provides a bias towards certain microbes? Question 3. What are the benefits and detriments of microbiome transmission between individuals, and do the benefits outweigh the detriments, or vice versa? So microbial transmission is usually talked about in terms of pathogenic bacteria, catching germs and things like that. But we also know that there are bacteria that can improve our health. Do you think that the potential risk of microtransmission is greater than the possible reward? Question 4. Is strain sharing an important variable to measure, and how can it be further studied? So the concept of strain sharing is fascinating, and it provides an insightful look at the movement of microbes from person to person. How do you think it could be implemented in different studies? And the last question, what ramifications could this study have on future approaches towards microbiome-associated diseases? Microbiome dysbiosis has been linked to gastrointestinal, neurological, and inflammatory diseases, as well as others. Do you think the findings of this study could be useful towards research into disease? And with that, I have finished my presentation. I hope you all have enjoyed this study and this presentation. I look forward to discussing this with you all, and I thank you for your time.